Hi, grade 7. Lesson number 1 in chapter 1 in biology book, Discovering Cells. What are cells? As we know, all things around us have a building unit, okay? As we know, the brick is considered the structure unit or building unit of the building. And atom is considered the building unit of the matter or element. Also, the cell are the basic structure and function unit of living organism. Whatever the living organisms, plants, animals, humans, or microorganisms. What's meant by microorganisms? Microorganisms are the tiny organisms can't see by naked eye, okay, but we can see them under microscope as bacteria, fungus, okay, virus. The cell contribute, okay, in the body function to keep the living organisms alive. What's meant by organisms' body function? Okay, organisms' body function are the processes that enable the living organism to live, grow, obtaining oxygen, food, and water, and getting rid of wastes and reproduce. Okay. Organisms' body do vital processes that keep them alive, and the cells in our bodies are involved in all these functions to keep us alive. We have many examples. The cells in your digestive system digest and absorb food, okay, to give the living organisms energy needed to do vital activities, okay, and the cells in your lungs help you to get oxygen. No one knew about the cells until about 1590. The invention of first microscope allowed people to look at a very tiny object can't be seen by naked eye, okay? So microscope is an instrument or is a device that makes small objects look larger by magnifying them, okay? So the main function of microscope is magnification of small objects or tiny objects, okay, to appear larger and show details. The history of the cell. In 1663, the English scientist Robert Hooke was the first one, okay, examine the cork of oak tree. Cork are dead cells inside the trunk of oak trees, okay? Robert Hooke used his microscope, okay? This is a Robert Hooke's microscope, okay? Used by him to examine the cork, okay? He saw empty spaces inside the cork. The empty spaces looked like tiny rectangular room, okay? So Robert Hooke named them cells. Cell is a Latin word mean a small room. After Robert Hooke, the Dutch businessman or the Dutch scientist in 1674, Leeuwenhoek looked at the drops of lake water and take scrubbing from teeth and gums and examine all of them under his microscope. Leeuwenhoek found a variety of one-celled organisms, okay? One-celled organisms, they are organisms, their bodies consist of one cell, and this cell make all the function, okay? Leeuwenhoek found a variety of one-celled organisms that move through water, okay? He called them animalcules, okay? Animalcules equal little animals. Leeuwenhoek In 1838, Schleiden, using his own research and other researchers, then conclude that all plants, whatever their size, are made of cells. A year later, Schwann reached the same conclusion about animals. So, all plants are made of cells, all animals are made of cells. In 1855, Virchow proposed that the new cells are formed only from existing cells. Okay? Their work and the work of many other scientists led to the development of the cell theory. Okay? The cell theory states the following. Number one, all living things are composed of cells. Again, all living organisms are made or are composed of cells. Okay? Number two, cells are the basic unit of structure and function in living organisms' body. Okay? Number three, all cells are produced from existing cells. Okay? The microscope helped us to discovering cells and unicellular living organisms or one-celled organisms and microorganisms. So how do microscopes work? Some microscopes focus light through lenses to produce a magnified image. Okay? Some microscopes depend on focusing light, okay, through a convex lenses. Okay? Convex lens, we know that the convex lens, a curved glass that thick at center and thin at edges. Okay? This convex lens used inside the structure of microscope, and the main function of convex lenses inside microscope focusing and gathering light to help the microscope produce a magnified image. But we have other types of microscopes. Microscopes don't depend on light, but depend on or use beams of electrons. Okay? By using some types of metals that emits electrons. But both light microscopes and electron microscopes combine two important properties, magnification and resolution. What are the difference between them? Magnification means the condition of things appearing larger than they are, okay? While resolution means shows their details, okay? Resolution of image depends on the power of magnification. If the magnification increase, the resolution of image will improve, okay? And when we obtain a better resolution, okay, we have more details, okay, about the things we are examining. This is image formed by light microscope, okay? While this is image formed by electron microscope. Electron microscope, okay, this is an electron microscope. In electron microscope, use a beam of electrons instead of light, okay? Electron microscopes allow higher or powerful magnification and better resolution when we compared them by the light microscopes. The compound microscope. Magnification by using a compound microscope. A compound microscope, okay, as we see, it's a type of light microscopes, okay? It consists of many parts. Number one, eyepiece lens, okay? Eyepiece lens. Number two, revolving nosepiece. This part called the revolving nosepiece. And the revolving nosepiece have three objective lenses, okay? This is part called arm. And this is part called stage, which has stage clips. This is part called adjustment, while this is part is a light source that emits light. And this is a base of microscope. Compound microscope magnifies the image by using two lenses together, okay? The magnification of compound microscope depends on two lenses that work together. One lens fixed, okay? One lens, as we see in the picture, one lens, the first lens is fixed in eyepiece, okay? And this is used by you to examine the slide. The second lens is chosen from a group of three lenses found on revolving nosepiece, okay? Each objective lens has different magnification, okay? So we can use the suitable objective lens to examine the slide. Again, we have two lenses, one of them fixed on eyepiece and the other chosen from objective lenses, three objective lenses fixed on revolving nosepiece. By turning nosepiece, you can select the lens you want. This is part called the stage. A glass slide, okay, you want to be seen, fixed or put on the stage, and it fixed by using stage clips, okay? Then, the light source. Light coming from a lamp in light source, okay, when we turn it on, passes through the object found on the glass slide, and then to the lower objective lens, then to the eyepiece, okay? So we can see the magnified image for the slide you examine, okay? The total magnification of the object equal the magnification of two lenses multiplied together. Example, if the lower lens magnifies the object 20 times and the base lens magnifies the image 10 times, so the total magnification of the compound microscope equal 20 times 10 equal to 100 times. Okay? Thank you. Grade 7.